The epithelium, a key component of the innate immune system, provides a physical and immune modulatory barrier. It acts as a border between the external environment and our tissues and functions to maintain homeostasis while defending against pathogens, pollutants, toxins, and allergens. This defense is provided not only by the physical barrier given by epithelial cell tightness, but also through antimicrobial peptide production, mucociliary clearance, mucus production, and permeability regulation. When the epithelium is damaged, pathogens, allergens, and other irritants can penetrate the barrier and induce innate and adaptive immune responses. A defective epithelial barrier has been found in many conditions, such as atopic dermatitis, asthma, allergic rhinitis, chronic rhinosinusitis, eosinophilic esophagitis, and inflammatory bowel disease. In response to stimulation by microbes, allergens, irritants, or cellular damage, epithelial cells release various cytokines termed alarmins, including thymic stromal lymphopoietin, or TSLP, interleukin-33, and interleukin-25. These alarmins are the body's ubiquitous warning signals following infection and physical or immunological insult. They are produced by both immune and non-immune cells and play a pleiotropic role in promoting the development of inflammation. Once released from the epithelium, the alarmins can activate and modulate innate and adaptive immune responses in overlapping but distinct ways. Alarmins are involved in both allergic eosinophilic, or type 2 inflammation, and non-type 2 inflammation, including structural changes. They trigger activation of dendritic cells, which initiate the differentiation of naive T cells into T helper 2 cells. Alarmins also activate type 2 innate lymphoid cells, referred to as ILC2s. These ILC2 cells, as well as Th2 cells, produce copious amounts of type 2 cytokines, namely interleukin 4, 5, and 13, and propagate type 2 inflammation. The development and maturation of eosinophils in the bone marrow is regulated by several key cytokines, including IL-5. IL-5 is critical for the differentiation, migration, activation, and survival of eosinophils. IL-13 and IL-4 act on the epithelial cells to induce mucin production through goblet cell hyperplasia and basement membrane thickening. IL-13, in conjunction with IL-4, is also responsible for proliferation of fibroblasts and airway smooth muscle cells that may contribute to nasal polyp formation in patients with CRS. IL-4 is also a potent inducer of the differentiation of T helper 2 cells, monocytes into M2 macrophages, and B cells into plasma cells producing immunoglobulin E, or IgE, that then activate mast cells, perpetuating the inflammatory cascade. Macrophages promote excessive cross-linked fibrin deposition and tissue remodeling, while IgE binds to the Fc epsilon receptor on the mast cell, activating release of its contents from secretory granules, including prostaglandins, leukotrienes, and histamine, as well as the type 2 cytokines IL-4, 5, and 13. This further amplifies inflammatory loops and sustains existing inflammation. Collectively, these cellular and molecular immune responses further contribute to the disruption of the nasal epithelial barrier that ultimately results in increased mucus production and water retention, increased microbial burden, and tissue remodeling that lead to the formation of nasal polyps. Type 2 inflammation, as just described, is responsible for much of the pathology associated with CRS with nasal polyps. However, non-type 2 inflammation also plays a role in a number of patients with CRS and includes neutrophilic and polygranulocytic subtypes of the disease. In fact, further evidence suggests that TSLP may play a role in non-type 2 inflammation by providing critical signals for T follicular helper cell differentiation, human B cell proliferation, and mast cell activation. As of early 2025, there are 3 FDA-approved biologic agents for the treatment of adolescents and adults with CRS with nasal polyps that can directly target the type 2 inflammatory processes. Unfortunately, there is still an unmet therapeutic need for biologic agents specifically indicated for non-type 2 inflammatory subtypes of CRS with nasal polyps. However, tezepelumab and itepekimab are fully humanized monoclonal antibodies that target the upstream alarmins TSLP and IL-33 respectively, and are currently being evaluated for their efficacy in treating CRS with nasal polyps. By targeting and inhibiting the alarmins, it is believed that not only can the type 2 inflammatory cascade be hindered, but also non-type 2 inflammation.